When we say silicified, what we mean is that silica has been dissolved in water and then come out of solution as that situation cools. Remember, we talked about silica being soluble in water at extremely high temperatures or under extreme pressure. And both of those conditions are actually pretty common in Earth's crust at not too deep. So when you look into nature and you see places where there are flint and chert, it doesn't really mean that there's been some kind of mineralization. It doesn't mean that there's been a chemical change that's produced those things. A lot of times it just comes in with groundwater. And that's really important for our story about fossilization because, of course, fossilization starts with an animal dying in the right place. Remember the pig. The second thing that has to happen is a flood. As is seen in this picture, you can see that the flood took out some places where there were once picnic tables, and you can see the sediment that was deposited. Oftentimes, there's quite a bit of sediment deposited, as in this picture that shows the results of a flood in China, where there was actually what looks to be a couple of meters of sediment that washed in as a result of this flood. Those situations are the ones that produce fossils. Now, we talked a little bit about calcium carbonate dissolved in groundwater, and it turns out that calcium carbonate is more common than silica. But silica is another mineral that, because of its solubility, will replace the non-mineral content of bone. So what do we mean by the non-mineral content of bone? Well, bone is a composite tissue, and if you look at it under a microscope, it looks like what you see in this picture. There's collagen, which is a complex protein, and hydroxyapatite, which is calcium phosphate, like I talked about in the last lecture. It's a mineral. And the composite that's formed is very strong. It's strong in several different ways. It has a high tensile strength as a result of the collagen, and it also has a resistance to bending as a result of that hydroxyapatite crystal. If you look closely at the fibers of these things, you'll see the physical components of them that make for this very strong composite. Now, the calcium phosphate, the hydroxyapatite, is resistant to weathering. It's a mineral. It's not going to break down. It's actually very similar to calcium carbonate in the way that it is preserved and is resistant to weathering and breaking down. The collagen, however, is not resistant at all to weathering, and under conditions that you find fossils, what happens is that the collagen breaks down as a result of natural weathering. The protein comes apart, and if you're at the right place, remember how we talked about there being a place where calcium carbonate comes out of solution in groundwater, or a similar circumstance for silica. What happens is that that silica-laden or calcium carbonate-laden water will seep into the bone and fill the space where that collagen fiber once was. That's what makes the fossil strong. The mineral part of the bone stays around. The non-mineral protein part is replaced by another mineral, either silica or calcium carbonate, and that's what produces fossils. This process, the process of mineral replacement through dissolution and replacement like we just talked about, is called permineralization. Some fossils do undergo metamorphosis, so you can find situations where the minerals that were replaced recrystallize, and these are more rare fossils, kind of more valuable. There's lots of different kinds of things that fossilize, and some things that fossilize you might not think of as fossils, but they are considered fossils. Things like footprints or leaf impressions, those give biological information about an organism that lived in the past. Really important fossils include things like diatoms and pollen grains. As you might imagine, there's quite a bit of ecological information contained in such things because oftentimes microorganisms or plant species have narrower ecological ranges, and that allows you to say a lot about the past, and we'll be talking about that in a little while. Stone tools are also something that you find buried along with fossils. In fact, once you get to that time when our ancestors start to produce tools, about 2.5 million years, stone tools all of a sudden become much more common than hominid fossils. Maybe not more common than all the fossils that you find, but they become pretty common. And so we don't really consider these things fossils, though they are traces of once living organisms. They don't really provide biological information. So that's what we're talking about when we're talking about fossils. let's use what we just learned to address a famous fake fossil, the fossil finger. Now, look at this picture. This is a supposed fossil finger. Now, if you've got your computer open and you're not Facebooking, and I've got my spies, so be careful. If you have your computer open, Google fossil finger, and you'll see that the first entry that comes up will show you this famous fossil finger. But now you guys are good enough to understand what we would expect from a fossil. If you accidentally lost your finger to, say, a shark attack or something like that, and your finger fell to the bottom of the sea and was buried in a condition that was amenable to fossilization, do you guys think that this would result? Probably not. And that's because most of the tissue of your finger is going to break down. If it wasn't the crabs and things like that that got it, actually the physical environment that normal fossilization occurs in reduces protein. It causes the weathering of protein to a state that is not something that would fossilize. So this is unlikely to be a real fossil. In fact, if you cut it open and look at the inside, it is most certainly just a normal siliceous, either a chert or a flint, that just happens to look like a finger. And actually, from knowing how to use an air scribe to clean a finger, I can see that a lot of that fingernail was actually doctored to look a little bit more like a finger. So this isn't really good information, and should I ask a question about the fossil finger on the exam, I would expect that you guys would be able to describe to me why this fossil finger is a fake. So now we're going to switch to dating the fossil record, because finding dates is actually very, very important in putting together a composite idea of what the fossil record looked like. So you can imagine that fossils and their context in time are a big part of what a paleontologist does. Now, if you remember, when we introduced the concept of fossils and deep time, one of the first things that people were doing is using fossils to detect what age the sediments that they were working in went back to. This was very important when you're doing coal prospecting or things like that, because it allows you to understand if you're in sediments that are too recent to really expect good coal, or other things like that that actually depend a lot on how long those sediments have been around. So the use of fossils to determine how old something is called relative dating. Now, even before absolute dating, even before the use of chemical radioactive means to determine how old something was, paleontologists had a pretty good idea of at least what came first. And remember how we talked about that stratigraphic column initially being separated in terms of what fossils were found in what places. So using fossils is actually one of the big ways, and this is called relative dating, where you're using the age of something not necessarily in terms of absolute years, but in terms of whether it came before or after something to put together the larger record. And we still use fossils in a lot of cases to date things, because knowing the evolutionary history and recording that evolutionary history with other radiometric dating techniques allows us to say that, for example, a certain horse group lived at a certain time in prehistory. And so when you are using fossils to determine how old something is, it's called biostratigraphy. And horse fossils are famous for doing that in North America. In East Africa, in the Pliocene and Pleistocene, oftentimes fossil pigs are used. Fossil pigs are really interesting in this regard, because what happens is there are a couple of lineages that go from being omnivores to being more herbivores. And as they make this transition to herbivory, their teeth get longer. And so in a couple of these lineages, you can see them starting out with small teeth and ending up with large teeth. This is something that in paleontology is really useful, because you can look at the length of that molar and you can tell pretty much how old that pig is. Now, absolute dating is when you can count the number of years. And technically, tree ring dating is absolute dating because you end up with an account for how old something is. It might seem like a really good method, and I suppose it is. The problem is that you need to be in a place that has a lot of trees. You also need to be in a place where it doesn't go back so far that you skip any time, right? So if you're looking at medieval churches in Central Europe, you have a pretty good chance of finding a timber in that church that can tell you pretty much when that church was built. However, if you're looking in the Pliocene and Pleistocene of the Rift Valley, the chances of you finding something that's going to fit any sequence is zero because there are no sequences that go back that far. So, good method, it just doesn't work in most circumstances where you find human fossils. Another method that's frequently used is called paleomagnetism, and this is kind of a combination of absolute and relative dating because we do actually know the exact times when Earth's magnetic poles flip-flop. The problem is that you can only tell whether a sediment is reversed or normal, so you need to have another gauge close to it to know whether or not that switch from reverse to normal is at one time or another, because there have been many as you go back in the past. Now, more commonly used to provide absolute dates are radiometric dating techniques, and these rely on radioactive decay. We've already talked a little bit about the argon-argon dating method, and we will be talking about that again. It's important to understand what we're talking about when we're talking about radioactive decay and when we're talking about the use of dating techniques to figure out how long something has been decaying. There are a couple of different minerals that commonly occur in Earth's crust that are useful in this regard. Some of them you probably have heard of for carbon dating. It's carbon and nitrogen. For argon-argon dating, it is argon and potassium. There's also U-series and uranium-lead dating. Now, uranium decays through a series of intermediate steps to lead, and it can be, in some circumstances, useful for radiometric dating. Unfortunately, when you're dealing with uranium series dating, oftentimes you have problems determining whether what you're looking at is an open system or a closed system. An open system is one where there's potential for, I guess, contamination. For example, in U-series dating, you're relying on the fact that you don't have any more uranium coming into the system that could change the result of your dates. Now, when you look at the different samples and the different kinds of techniques that are applied, you see that there are techniques that are commonly applied to younger samples and techniques that are often applied to older samples. It turns out that the argon-argon or potassium-argon techniques are utilized on relatively old samples, and this is probably the most useful of all the different dating methods in paleoanthropology. One of the issues with it is that unless you have giant crystals that can trap a lot of argon, you really have a problem with having enough argon to even measure because it doesn't build up all that fast. So if you have things that are younger than about 100,000 years, you better have some pretty darn big crystals. Now, it doesn't mean it's impossible. It turns out that the Pompeii eruption that we can record in history and see how old it is just from historical means had some pretty giant crystals, and potassium-argon dating actually nailed the Pompeii eruption. Now, another technique that's often used is the uranium-lead technique, and the uranium-lead technique is one that actually requires a circumstance that isn't really very common in Earth's crust. It requires a mineral that you don't see very much. So while it's a pretty accurate or pretty accurate dating method to be used on materials if you have them, it doesn't work for everything. So there's also a lot of less reliable dating methods, fission track dating being one of them. electron spin resonance. There's a whole bunch of these, and I actually don't require that you guys know them because it's not all that useful. In some cases, people really, really want to get dates, and so what they will do is they will use some dating methods that are actually known to be a little bit sloppy when it comes to not being a closed system. They're open system. So I'm not really even going to present those to you. There's also a type of dating which allows you to associate a particular set of sediments with a particular phase of global climate using the oxygen isotopes of the ocean. We'll talk about that in a little while. Now we're going to talk about those specific dating methods in a little bit more detail to give you some background. So the first one that we're going to go into in detail is what's known as the uranium-lead dating method. And this dating method only really works on one kind of thing, zircon crystals. And these zircon crystals are not found everywhere in the world. In fact, they're pretty uncommon, it turns out. But when you do find them, they can be dated. And it's this dating method that was the first one to show how old the Earth was. This dating technique showed that Earth's age was in the billions of years. And as you'll see in the reading that you do for your paper next week, this is something that actually had a major effect on biology because up to that point, people really didn't have a good handle on how old the world was. You remember from previous lectures that they were using the thermodynamic properties of the Earth to try to calculate back how far it had been since the Earth had cooled from a molten state. And these methods really didn't work so well. By the time that you had the turn of the century from the 1800s into the 1900s, people had become a little bit more amenable to hundreds of millions of years. However, it wasn't enough time to really give all that evolutionary process the time it needed to build the tree of life. And so there were a lot of people that were saying that there just wasn't enough time that was in existence to account for the tree of life. So when people first started to find these datable zircon crystals in infrequent sediments, they were able to date them and show that Earth's age actually was in the billions of years. So here's a zircon crystal, and the thing about zircon that is really cool is that in this crystal structure, it readily incorporates uranium and strongly rejects lead. But then once you get to that crystalline structure, of course, once that crystal is formed, uranium is well known to be a radioactive substance, and it starts to decay. And it decays in a well-known sequence. It's multiple steps, but it goes through these steps at known rates as it breaks down into lead. And so because this zircon has rejected the lead in the crystal formation, by measuring the lead and measuring the uranium, you can see how long ago this zircon crystal formed. This is a series of the different steps that uranium goes through on its way to becoming lead. And so it's called uranium lead dating. U-series refers to a similar process, but it's a little bit different. We'll talk about that in a second. Uranium lead dating does have a few problems. The first one is that zircon is highly stable, and so it retains the lead that's generated by uranium even in very hot temperatures. So you've got to be sure that the sediment that you find those zircon crystals in is the place where they actually crystallized, or else you're just dating the zircon crystals and not the surrounding sediment. You have to be pretty careful to make sure that the zircon being tested originates in the geological feature that you're trying to date. A bigger problem is that you just don't find zircon everywhere. It's really uncommon, actually, and so it's something that you don't really see in too many places. Now, this dating technique, uranium lead, can date really, really old events because of the long decay scheme of uranium to lead. In fact, the oldest known rocks on Earth have been dated using this technique to about 4.5 billion years. Uranium lead dating is usually used to date minerals that are older than what's relevant to human evolution. And like I said, they're just not found with most stuff that's relevant to human evolution. Okay, so we've gone over this one before. I'm going to go over it slowly again right now. What shoots out of a volcano is not smoke. It's a combination of a couple of things. Most of it is ash, and that is quickly cooled silica. And that comes out looking like pumice. We've seen that before, and quite a lot of material can be ejected from a volcano. You'll remember that there is also other stuff that comes out of a volcano. We'll get to that in a second. But for now, know that during major volcanic eruptions, which are pretty frequent in a geological context, from a geological perspective, they're pretty frequent, quite a bit of material comes out. Some of that material falls, lands, and is deposited where it lands. This is called airfall ash. Other material gets caught up in some fluviatile lacustrine system and is deposited by water. Obviously, ideally, you want the material to be transported as little as possible. An airfall ash is perfect because that means that that deposit was laid down at about the time of the volcanic eruption. Those are the best case circumstances for dating. Most of the time, there is some water movement. And in a lot of cases, you can deal with that also using the argon-argon dating technique, which I'm going to elaborate on a little bit further and explain to you why it is that you can use that technique even if you don't have absolute certainty about whether or not this tuff was laid down within moments of the eruption. Now, airfall ash is perfect. Generally speaking, they are transported by water to a certain degree. This picture of a tuff has been transported, and you can see that by the layering that occur in those sediments. When you look at what comes out of a volcano, like we talked about, there is both the material that comes out as cooled silica, so little pumice shards, little glass shards, and that's most of it. And then there's crystals, right? And there's a bunch of different kinds of crystals that come out of volcanic eruptions. Basically, those crystals that don't melt at the temperature of the volcanic eruption will stay in the lava as is, right? They'll stay in the lava and the ash as long as they don't have a melting point that's lower than the temperature of the volcano. Now, like I said, there are a lot of crystals that fill this role. Okay, so there's a lot of stuff that comes out of a volcano. Most of it is ash. There are a lot of different kinds of crystals. There are certain classes of crystals that are composed of molecules that contain potassium. And these are the ones that are important for the argon-argon and potassium-argon dating methods. You remember I made the comparison of a big quartz crystal with some milky imperfections inside of it. Those imperfections are disruptions in the crystal lattice makeup of that quartz crystal. So the crystalline matrix of the quartz has places where the crystals, the sequence of molecules that make the crystals, are kind of separated. Gas can get trapped in those. And in the case of certain groups of minerals, like potassium feldspars, sandines, there's a few other ones, potassium-containing crystals. In a lot of cases, gas can get trapped inside of those imperfections. Now, when the crystal contains in its molecular structure potassium, that is really important because potassium occurs in actually several different forms. One major form that's not radioactive, and a second minor form, it's little more than a trace, but it's radioactive. And that radioactive version breaks down into an isotope of argon. Argon is a material that then gets trapped in that crystal lattice of the sandine or feldspar crystal. Now, when that crystal is heated to the temperature of volcanic eruption, it allows the trapped argon gas to escape. It essentially resets the crystal. It sets it to 0 argon. Therefore, any argon that starts to collect in those imperfections happened after the eruption. And it starts to happen as soon as that crystal cools off. So the eruption occurs, heats up the crystal, allows the argon to escape, shoots that crystal out, and the crystal gets deposited in an airfall or other type of ash, and then the argon starts to build up in the crystal again. And we know the decay rate, we know how long the half-life of that radioactive isotope of potassium is. It's 1.2 billion years. It's a long time, but it allows things to be measurable. It allows for older sediments to be measured pretty readily. Readily, that is, as long as those sediments contain volcanic tuffs that have crystals that trap that argon. And like I said, there's a whole range of them that frequently occur with volcanic eruptions. Okay, so how do you do this? Well, you have to measure both the potassium that originally exists, and you've also got to measure the argon, right? So you have to measure the amount of potassium that occurs in that sample, in that crystal, and then you have to measure also the amount of argon that occurs in that sample. And it's a specific isotope of argon that you're measuring. The argon-40 is the one that we understand the decay sequence of. So the decay of the potassium-40 into argon-40 allows us to understand how long it's been taking for the argon to decay. Okay, so to get a date, you need to know how much of that radioactive potassium occurred in the first place. And you measure it using a mass spectrometer. Now, unfortunately, you cannot measure both the potassium-40 and the argon-40 in the same mass spectrometer reading because there's different ones for observing potassium and for argon. What that means is that if you're using a traditional potassium-argon dating method, you need more than one crystal. This is problematic for anything but an airfall ash because water might incorporate crystals from more than one eruption. So what you need in order to date a single crystal is a single mass spectrometer reading. And that's what the argon-argon dating method is. What you do is you send it to a laboratory that has the capacity to irradiate the crystal, turning the potassium-40 into a different isotope of argon, argon-39, at a known rate. So it converts the radioactive potassium that occurs in the crystal into a known amount of argon-39 based on how long it's been irradiated. You can then take that crystal that's been irradiated and has the argon turned into, or the potassium, has the potassium turned into argon-39 and measure that argon-39 in the same mass spectrometer reading as the argon-40, allowing you to use a single crystal and a single mass spectrometer reading to obtain your comparison of the original amount of radioactive potassium, potassium-40, to the argon isotope that you're interested in, argon-40. So it allows you to date single crystals. And why that's important is because in a tuff that's been transported a little bit, you will have crystals that all have the same date. In a tuff that's been transported a lot, you might have crystals with vastly different dates. Imagine if you have a tuff that goes back to the Miocene, earlier than 5 million years, combined with a tuff that you think is about 300,000 years. Using the traditional potassium-argon dating method, you would have a problem because you wouldn't be able to be sure that that crystal is from the same eruption, right? So using the argon-argon dating method and looking at a broad range of crystals from a particular tuff can allow you to date a population of crystals. So why is dating a population of crystals important? Well, imagine that you have a tuff that's transported by water and you collect crystals out of that. Maybe you don't know how far it's been transported by water, but if all the crystals that you pull out of that tuff have the same date, you can be pretty sure that you're dealing with a single event. And so those streams that were choked up by the Mount St. Helens volcanic eruption, those major ash flows and streams that occurred right within the couple of years proximal to the Mount St. Helens eruptions we'll have populations of crystals that all date to the same volcanic event. And even if they have been moved 10 years, on a scale of 1,000,000 years, that's not a very big deal. So it ends up being a very, very valuable amendment to the potassium argon method. Now, the potassium argon method, as you'll learn as you do your reading for the paper that's due next week, is something that was developed here in the Bay Area in Berkeley. And you'll be reading about the guy who did it. And it has a very profound effect on our understanding of prehistory and human evolution. As you'll see, it occurs at the same time that we start to realize some pretty major things about human origins. One, that the African apes are probably a little bit closer to us than orangutans or gibbons. Much more importantly, by dating things, we were able to show conclusively that Homo sapiens is a very young species. And all these crazy projections for a long depth of time in separation of all the different people that live here were wrong. There hasn't been very much time separating all the humans at all. In fact, it's been a very small amount of time, and that's why we don't think of different groups of people, different so-called races, as being different lineages. Obviously, they're not. And by using the potassium argon dating method and framing up the human evolutionary record in terms of time, we were able to show that Homo sapiens is a very young species, and that these notions of some deep divergence between different groups were not based on reality. You'll understand why that's important. Let's next look quickly at radiocarbon dating. It turns out that this dating method isn't used very much in human origins research either. It's used a lot in looking at early modern Homo sapiens and also Neanderthals, but it really only works back about 50,000 years in ordinary circumstances and maybe a little bit longer than that in very exceptional circumstances. But because the half-life of carbon-14, the radioactive version of carbon, is so short, 5,730 years, we really don't have that much time before we end up reaching the limit of what we can measure in terms of available carbon, right? So if you go too far, if you go too many half-lives back, if you go 500,000 years back, there's such a small amount of radioactive carbon still in the sample that you really can't hope to measure it and compare it to the nitrogen. Okay, so how do we do carbon dating? It's actually much simpler in a way because it's more intuitive. So first of all, nitrogen in the atmosphere is converted to radioactive carbon in the upper atmosphere. It's bombarded by solar and cosmic radiation, and so carbon-14 is produced all the time, and it occurs in a steady ratio with the nitrogen-14. Now, carbon-14, just like any radioactive isotope, will form into the same molecules as its non-radioactive cousin. Right? So carbon-14 will be incorporated into carbon-containing molecules just like non-radioactive carbon-12. And so the carbon that composes living things is made up of some component of carbon-14 and some component of carbon-12. In fact, the amount of each of those things is determined by the ratio of carbon-12 to carbon-14 in the atmosphere, which is more or less stable over time. We understand the history of carbon-14 in terms of these ratios. And so we can measure the carbon-14 and compare it to the carbon-12. And that is something that, if it's a living thing, is going to occur in the ratio that exists in the atmosphere. Now, once that animal dies and it stops interacting with atmospheric carbon, it's a different story because that carbon-14 immediately starts to decay, and it decays back into the nitrogen-14. So what that means is that over time, things that were once living but not living anymore will build up their ratio of carbon-12 versus carbon-14. By comparing the ratio of carbon-14 to carbon-12, therefore, you can tell how long it's been since that living tissue was interacting with atmospheric carbon. That's how carbon dating works. Again, it really doesn't go back all that far. Now, the next dating method is the U-series dating method, and this can be really useful under sort of exceptional circumstances. Now, it turns out that the substance that gets dated with U-series dating is actually pretty common. Calcium carbonate nodules are what substance you're using to date, right? And those occur pretty frequently. The issue is that in many cases, this is not a closed system. So, do you remember that picture of the groundwater produced calcium carbonate nodules, pedogenic calcium carbonate nodules? Well, that's what you look for when you're using the uranium series dating method. And what is unique is that groundwater contains dissolved uranium but not dissolved thorium. Right? So uranium is going to decay to thorium. And so if you can find a nodule that was precipitated out of the groundwater and then closed off, you have a chance of dating it. However, there is a little bit of an issue because it's not necessarily a closed system. Oftentimes, anywhere where you have groundwater and a fluctuating water table, what will happen is that those calcium carbonate nodules will go through several cycles of dissolution, and they might get soaked and get sort of reinvigorated with uranium from the water. And so you have to be very careful when you're finding circumstances that are good for uranium series dating. Remember that it goes through all these different steps on its way to thorium. And so with U series, what we're looking at is comparing the amount of uranium to the amount of thorium-230, basically. Now, what you look for is very large nodules and then go for the very center of those large nodules. Of course, that's the place where you're going to have the least interaction of that calcium carbonate nodule and the groundwater. Now, as you might imagine, that's not always the circumstance. And so we really have to be careful about the uptake of uranium that occurs after that calcium carbonate nodule has been formed. It's a big issue, actually. Yes, a big issue, but it actually is still a very useful dating method because sometimes the conditions are right. And when the conditions are right, you can date things that are kind of right in between that upper end of carbon dating and the lower end of argon dating. So, stuff when you're dating with the argon method should, generally speaking, if you have normal sized crystals, be 100,000 years old or older. When you're dating things with carbon 14, they can't really be much past 50,000 years. So, this period of time between 50,000, 100,000, 200,000 years is often what people who are using the uranium series dating method focus on. Now, this is a method where you really have to trust your geologist. You really have to be careful to select a geologist that is diligent about what they do. And that's an issue, right? That's actually an issue. You have to be very careful that the geologist samples the calcium carbonate from the right place. There have been uranium series dates that get checked in multiple labs and it works out. So the method does work. The problem is that whether the system is open or closed is a determination that must be made in the field, and therefore you need to really trust your geologist. Remember when we were talking about peer review and we were thinking about the potential problems with peer review? Well, here's one of them. How do you know, as a reviewer of a paper, whether or not the geologist sampled the rock from the right place? That's not a test that you can easily do. Now, science will self-correct, but oftentimes the difference in time from one group working in an area to another group working in an area can be decades. So you can imagine that in those kinds of circumstances, the self-correcting aspect of science can be kind of slow. From your guys' perspective, it just means that you got to be careful about uranium series dates. And, when you read them in a paper, it doesn't necessarily mean to discount them, but you would be happy if there were numerous uranium series dates done by numerous different teams and they all checked out. When you see different teams getting strikingly different uranium series results, that's a time to really be worried. So once again, U series is utilized a lot because it ends up dating stuff that are in between carbon dating and argon-argon dating. It's just sort of problematic. Okay, we're going to shift gears a little bit here and start talking about climate change. Climate change is something that all of you guys have heard kicked around here and there, and it's something that's pretty important because we have a little bit of evidence of carbon increasing in the atmosphere. In fact, we have quite a lot of evidence for it, and we also have evidence of global temperatures rising, and there's some suspicion that carbon spike might be related to increases in global temperatures, which, of course, as you know, have all kinds of consequences. Now, to tell you the truth, we're not really going to be talking about that kind of climate change. The kind of climate change that we're going to be talking about is stuff that's been happening for a long time. And, of course, that's one of the problems with proving that humans are causing climate change. The climate has been changing at various different levels, at various different time frequencies, for as long as we can measure it going back. There are reasons that we sort of know about that we associate with this climate change. The first is that the Earth's axis tends to tip. And it's kind of interesting if you think about it. Imagine an Earth that was tilted at 90 degrees, right? An Earth that was tilted at 90 degrees would actually have a pretty major buildup of ice. An Earth that was tipped not at all would have a lower buildup of ice. It's kind of interesting when you think about it. So the more that the Earth tips sideways and points only one side towards the sun, the more that ice builds up during the winter. So imagine the Earth is still spinning around the sun, tilted at 90 degrees. Half the time it's freezing up, half the time it's thawing out. It turns out that the freezing will hold through the thawing all through the summer if it's tilted at that 90 degrees to such a degree that the ice will continue to build up. Whereas when Earth's axis is at a straight up and down, it won't build up as much. Now, of course, the Earth never tilts so that it's 90 degrees on its axis, but there is a little bit of a wobble. And that wobble is one of the things that is the most associated with ice ages. There's a couple of other things, like the equinox actually changes every year a little bit, and so over the years you'll actually get sort of a different part of the Earth tilting towards the sun at different times of years, and that will have an effect. The other thing is that an ellipse actually has a greater average distance from the center point than a circle does, and so that will change as Earth's elliptical orbit changes. So these three different processes are all known to be operating at different time intervals. The most important of these is the obliquity wobble, because this is what leads to ice ages. When you tilt the Earth's axis a little bit farther than normal, you have a buildup of ice at the polar ice caps. Now, ice ages are interesting because actually they're only ice ages in those places that get affected by the polar ice buildup. Around the equator, you actually don't have any difference in the temperature, right? Because there's really not any other sort of distance from the sun thing going on. It's just a matter of the ice building up on the ice caps as a result of the colder, more intense winters, right? The summers are hotter too, but it just doesn't melt the ice. So when you're talking about ice ages in the past, it's mostly as a result of that obliquity wobble. Now, if you think about it, when there's more ice buildup, that means there's more water that's taken out of the atmosphere. right? Because there's the precipitation cycle where water is evaporated from the ocean and then is rained down or snowed down on the land, eventually making its way back to the ocean. If that water never makes it back to the ocean, then it stays out of the cycle. And it turns out that that phenomenon is exactly what we use to look at paleoclimate records in the past. We have a very, very good sample of cores from ice in the polar caps and from sediments in the bottom of the ocean that slowly built up that allow us to look into the past and to see when ice ages were occurring. The way that's done is by using another isotope, an isotope of oxygen. But we're not really worried about radioactivity in this case. What we're worried about is that oxygen 18, when it combines into a water molecule, produces a slightly heavier water molecule, and it tends to build up in the depths of the ocean, less than at the surface, where there's a higher percentage of normal oxygen 16 in the water molecules. And what this means is that during ice ages, that oxygen 16 is preferentially evaporated and snowed. And so the oxygen 18 occurs in higher concentration in the ocean during ice ages. It turns out that we have a very good record of this. Those drill cores that I showed you a slide back are located in lots of different places in the ocean, and they all tell kind of the same story about this cycling of oxygen 18. In fact, they tell such a good story that oftentimes you can detect differences in sediments on land that allow you to associate those sediments with a particular period of oxygen 18 buildup. Okay? Another form of dating is by relating a past sediment to a particular oxygen 18 isotope stage because they've occurred regularly in the past, and we know very well the climate background of Earth's history. We know it so well, in fact, that we tend to overemphasize it. It turns out that the paleoclimatic record is way better than the fossil record. And very often that climatic record is something that makes us think that climate has a lot to do with the way things went down in the past. In fact, there are a number of things called climate forcing models. It's kind of not a real well-known term, but it's a way of talking about models of evolution that rely on climate for their explanation. I'm going to introduce you to one of these. One of the most well-known hypotheses about how humans came to being. In fact, a lot of you guys might have even heard this, is called the savanna hypothesis. This is the idea that the, generally speaking, move towards cooler and drier climates that has occurred. And when I say cooler, it means drier, because that means that you have a buildup of the polar ice caps, which is taking water out of the precipitation cycle. So I said that Africa, equatorial areas don't really get colder during ice ages. They do get a lot drier. And that's the way that we can actually relate these on land sediments to the oxygen isotope stages. In any case, when you're with the savanna hypothesis that you guys have probably heard about relates to how humans emerge from this environment. The savanna hypothesis, which was first proposed in the 1920s when people first started to find ape men from Africa, goes something like this. Human traits like bipedality evolved as a result of the breakup of closed environments in Africa that required people to walk greater distances, and walking would seem to be more efficient than a different form of locomotion and would also potentially allow humans to carry stuff. So Bipedality, big brains are the consequences of adjusting to this new type of environment that was existing as the climate changed to one that was relatively cooler and drier. Now, these things are problematic because they're based on correlations and not necessarily a good idea for causation. Right? So, yeah, the climate is getting cooler and drier as we go across the Pliocene and Pleistocene, and that probably does relate to increases in the amount of savanna and decreases in the amount of tropical rainforest. But do we really know that that is the causal factor that caused us to become bipeds and have large brains? To be honest, most people, as they're first starting to understand evolution and adaptation, generally think that most adaptations are a result of animals adapting to the physical environment. That's not always the case, however. In fact, it's probably usually not the case. We don't really know, but we can say for sure that there's much more than the environment at play. Imagine what are the major selective factors against humans these days? Well, it's sort of the environment, but honestly, it's things like disease. If you're thinking about us in the Pleistocene, it's also things like predators. There are a lot of things that can affect your reproductive success besides just how cold it is or how rainy it is. Many would say that it's most likely other living things that are actually affecting you more than the physical environment. This is called the Red Queen hypothesis. And the reason why it's called the Red Queen hypothesis is because from Alice in Wonderland. In Alice in Wonderland, actually from the book Through the Looking Glass, the Red Queen has a famous quote. Alice is running and she doesn't seem to be getting anywhere. And she asks the Red Queen, "What can I do? I'm not getting anywhere and I'm running just as fast as I can." And the Queen looks at her and laughs and says, "Now, here, you see, it takes all the running you can do to keep in the same place. If you want to get somewhere else, you have to run at least twice as fast as that." And basically, that refers to the fact that in nature, because of the competition between organisms, because of the competition between organisms and parasites and diseases and things like that, you can't just stand still because you have to be running just to compete with the other stuff that's there. So it's not just about the climate, it's about you're competing with all of the other things around. For example, think about the lion and the hyena in this picture. The hyena is not worried about whether it's cold or not at night. It's worried about whether it gets any food and also whether it gets snagged by a lion, right? That's the Red Queen in action. It takes all the running that you can do just to stay alive. If you want to go anywhere, you have to be lucky, right? So it's probably not the case that the climate is the only thing driving human evolution. In fact, it's very likely that competition with other animals or just competition with other humans is actually in large part driving our evolution. And at least it means that we can't just look at the climate, look at the changing climate, and then relate all the different things that happened in human origins to that climate. We can't do that at all. In fact, it becomes very difficult to sort the different things out that are at work on human evolution. Now, we'll be getting to this more and more as we get into the class, but one of the things that is really interesting about adaptation and evolution as you get further into things is that there are a lot of questions that can't really be that easily answered. And when you're looking at something like bipedality, there's a lot of different hypotheses for how bipedality might have come about. And you need to weigh all of them, and we might never be able to know for sure which of the different possible factors was the biggest one, if that even really is a sensible way of looking at it. So at the end of our climate and geology lecture, we're left sort of wondering what it is that we can think about when we're thinking about what drives human evolution. Okay, here's a little intermission for you guys. Go ahead and take 5. We'll be back to a lecture in 5 minutes. So have a good fractal. Okay, I'm sorry. Party's over. Back to the boring stuff. It's not that boring, actually. It's the only story about your past that's real, and it ends up having all kinds of real effects on how we interpret day to day life. So, yeah, I guess you could think of it as boring. What the heck? It's boring. It's more boring than listening to music and watching fractal videos, anyway. But we've got to do it, and that's what we're here for. So now we're going to start talking a little bit about, I guess, macroevolution is a good word for it, although we're not really going to get into too many of the details. If you want that class, I'm teaching it in the fall. It's 3100. It's a great class. I'll be touting it a little bit more when I come back and do it in person. But from the perspective of a human paleontology class, basically what you need to be aware of is how species come about. And you need to be more aware of sort of the history of how people understood species. Because it turns out that at the time of Darwin, nobody really got what species were at all. They just thought they were these fixed entities that didn't change at all. And when Darwin came up with his hypothesis for how species originate, remember, origin of species, the big deal is that species do originate rather than natural selection. Natural selection was a concept from before, but how species originate is a really big deal. And the way that Darwin conceived of it was that species originated through what's called allopatric speciation, that the species exist as a result of being separated by geography. Like we talked about a little bit before, that's not really the case. We know that species can actually form when the two organisms are living together in the same place. We use the polar bear as an example, and you'll see we'll use that as an example again here. In any case, this process of speciation is really important when it comes to understanding the human fossil record, because there's a whole bunch of species names that get thrown at you. And unless you're kind of aware of what species are, these can be really, really confusing. In fact, even if you do know what species are, they're pretty confusing. And that's because of the rules of nomenclature. It turns out that the rules of naming species are sort of different from what we know species to be. Now, I'll be back in person to explain that one in the next half of this lecture. However, we'll start the ball rolling with a little bit of history. It's the only real way we can do it. And we'll also start with a few basic concepts about what a species is. Have you guys ever thought about what a species is? It's not that simple, right? Of course, if you're walking through the forest, it's the different kinds of things that you come across, but you know what? If you look at coyotes in the LA basin, they're full of dog DNA. Does that mean that dogs and coyotes are actually the same species? It turns out that there's big hybrid zones between wolves and coyotes, and those two animals have been separated for at least 1,000,000 years. Well, sort of separated, except in the hybrid zones. It actually turns out that when you look in nature, and especially if you put similar animals that occur in separate areas in nature in captivity together, they can often interbreed. So, this definition of species, like strict biological species that might not interbreed, that cannot interbreed, actually, a lot of the things that you might classify as different kinds of animals and plants wouldn't fit that definition of a species. So, what do we use? Well, I mean, fortunately, it turns out that in nature, a lot of times animals that'll breed readily in captivity don't interbreed. And so the biological species concept, that is, this notion of interbreeding, defining what is and what isn't a species, works pretty well, just not perfectly. So here's a few examples of different animals that you might come across that are not the same species, but they'll interbreed in captivity. Things like ligers, things like different breeds of horses. Of course, you guys know about mules, but do you know about zonkies? Yeah, zebras and donkeys can interbreed. It turns out they do a lot better than horses and donkeys, or zebras and horses. They'll also interbreed. They produce sterile offspring. The donkey zebra does not produce sterile offspring. Did you guys know that your ordinary average house cat can interbreed with pretty much every other small cat that's out there? This is a savanna cat. Savanna cats are a cross between a domestic cat and a serval. And yes, they are legal. You can find them in some places, in some pet stores. Wolfotes, growler bears, camels, and llamas can interbreed together. So it's actually pretty common that you have these potential hybrids, and it makes you really wonder what species are. But at the end of the day, when you do go walking through nature, the species are real. They're real, right? They're different things that you come across. And so it is useful to classify those things. But it makes it really tough when you're trying to apply these concepts to the fossil record, of course. So it turns out that in biology, there are a lot of different ways to recognize species. You can recognize them based on whether they can interbreed. You can recognize them based on whether they do interbreed in nature. Ever thought about asexually reproducing organisms? Things like single-celled organisms, or actually there's asexually reproducing vertebrates also, where there's just essentially all females. What do you do with those? Obviously, the biological species concept isn't going to work because mostly new forms are made without mating at all. So, it's kind of interesting when you start to get into all the different possible ways that you could define species in biology, and there's a lot of different ways, but really it comes back to first the different kinds of things that are out there, and second, some semblance of a biological species concept. Now, this sort of thinking is really, really important when it comes to the human fossil record, because while there are lots of human fossils, generally speaking, those fossils are kind of scrappy, and when it comes from any particular site, usually you don't have that many individuals. It's very rare, actually, that you have more than one individual. And there are some special cases where there are up to 17 individuals, well, depending on what you're talking about, the minimum number of individuals or the number of crania. There's a site that has 17 crania, and you can say something about variation and everything like that, but usually it's not like that. And what that means is that species are difficult to deal with in human paleontology. They're named based on rules that are set forth by the International Code of Zoological Nomenclature, and they're related to classifying the organisms. When you use evolutionary pattern to design the taxonomy is just the words that you use. And when you use evolutionary pattern to try to make sense of the taxonomy so that the taxonomy, the names, correspond to evolutionary pattern, that's called systematics. And so when we're doing systematics, we're using evolutionary pattern to name species. But of course, it's difficult because, at the end of the day, the thing that we really kind of go by in our gut is whether organisms reproduce with each other, whether they can or not. Nobody would ever put a chicken and a horse in the same species because they couldn't possibly reproduce together. Most people probably wouldn't put a dog and a coyote in the same species either, but they can reproduce together when you have exceptional circumstances. So anyway, this all goes to show that species concepts are difficult in paleontology, and human paleontology in particular, and history, it becomes really, really important. So quickly back to our taxonomy and systematics slide. One of the things that you'll be responsible for is genus names, and one of the names you'll be responsible for is species. Now, these are the two lowest levels, the two lowest common levels within the Linnaean hierarchy. And that means that species has a sort of particular kind of definition. We've already started to talk about these different concepts of species, but what about genus? Well, genus is something that's a little bit less well defined. You can't define it based on reproduction at all, of course, because there are plenty of different species within a genera, and they don't have to interbreed to be in the same genus. So genus names are kind of tricky, but of course we have to kind of think about those when we're thinking about the human fossil record. We'll talk about that when we get sort of further into things, but one of the things that you do need to know is that most often when you refer to an organism in scientific terms, you use binomial nomenclature, which means that you use a genus and a species, and they're both always italicized. They're the only ones in the whole Linnaean hierarchical classification that are italicized, but genus and species are both italicized, and genus is capitalized. And so something like Australopithecus afarensis would be written with either an underline or italics and then a capital letter for Australopithecus. So to understand the way that species apply in human origins, like I said, we have to get a little historical. We have to look at the history of discovery, because when you name a species, the way that the process works is that whatever species name is first applied, that's one you've got to use for that whole group. As long as you align a fossil within a group, whichever fossil was first named in that group takes the species name priority. And so it's really important to understand history, because, of course, the earlier a species was named, the more likely that it's going to be around. And the very first fossils of humans, of human ancestors to be discovered, or actually, as we'll find out, not really human ancestors, more like cousins, but there's a little bit of Neanderthal in all Eurasian and North American, South American Homo sapiens, which are essentially Eurasian Homo sapiens. However, that amount is negligible. Anyway, the first Neanderthal cranium was discovered in 1856, and when it was first found, people just thought it was weird, and there was a lot of debate about actually about what that thing was. A lot of people suggested that it was just a weird-looking human, and to be honest, the cranial capacity isn't any smaller than a modern human, so it really was something that was really plausible. And it was at a time when people were only first starting to take pictures, and there were a lot of stories about the crazy Cossacks who had come in and fought during the Franco-Prussian War, and there's all kinds of different conceptions that maybe there's some weird-looking group of people out there that might belong to this skull. So nobody put it in its own species at first. It took a while before it got named a new species, and it actually took until after Darwin wrote The Origin of Species and people started to contemplate this notion of evolution by speciation. And then people did name it, right? So there's a big debate at first, but eventually we do get a name for this Neanderthal specimen after Darwin writes The Origin of Species, and after at least the intellectual parts of progressive Europe and Britain and the Americas had started to digest what Darwin said in his book. And it was a sort of a hard thing to digest. And as we'll see, the difficulty in understanding that and humanity's sort of perception of itself as unique has done a lot to prevent the learning about what we are actually, and it's done a lot to prevent us from seeing things how they really are. One of the biggest issues that humans had with evolution, the way that Darwin talked about it, is that inevitably our closest relatives in the animal world were apes and monkeys, and nobody really liked this very much. And so we see, and actually we continue to see it today, but not really in science since the modern evolutionary synthesis that we talked about before. Not since then have we really seen this kind of thing in science, but there was, at least from the publication of The Origin of Species until the discovery of ape men in Africa, a lot of resistance to this idea that when we looked into the human past, we were going to see things that were progressively more ape-like. But it's almost like it was a debate between two different sides, because it wasn't too long after Darwin published The Origin of Species that Neanderthals were named as a separate species, and their ape-like features were actually emphasized in this species naming. And so at sort of right away with that publication, there's almost like this implied hypothesis that Neanderthals are the ancestors of Homo sapiens. This is another thing that is going to take a long time to work itself out of the literature for science to self-correct. Things took longer before. The internet really speeds things up, but this was a big issue. So Neanderthals are proposed to have a new species name, and they are also proposed to be the direct ancestors of humans. Now, and their ape-like features are emphasized. Uh, One of the ways that people dealt with this that we've already talked about was to suggest that these are actually just weird humans. So that's kind of how the debate was uh, kicking around for the first 2 or 3 decades from the discovery of the Neanderthal cranium. But then another big discovery at Spee, it's spelled spy, but it's pronounced Spee, in Belgium showed multiple individuals with the same morphology. So very important because it means that this is not just some weird looking modern human. These are a real different group of things. This notion that you could possibly have a uh, just a weird looking human didn't make sense anymore in the context of what were now three of these individuals. Some from the same place, and they all look the same. They've all got the big brow ridges and sort of the low vault, and they all have these same seemingly primitive features. So, just at the same time that you're starting to get people realizing that these Neanderthals must be something real, a true missing link is discovered. Now, Neanderthals have brain sizes that are about the same as modern humans, if not a little bit bigger. But the first real missing link is found in the late 1800s, 1891. It's a Homo erectus specimen, and it's found in Indonesia. And it is truly different from anything. Now, if you look at it, it kind of superficially looks like a Neanderthal, but it's a lot smaller. It's a lot smaller. And one of the ways that this new human fossil is interpreted, it turns out, well, a lot of people just didn't even want to accept it as real. There were actually the majority of scientists that line up against it and kind of don't want to accept it. But some people are starting to think of this, what's called unilineal, like a single line of human evolution that includes this early missing link, Homo erectus, along with the Neanderthals and humans. And this idea is, it was a minority opinion. It wasn't something that most people who were in the field thought. And part of the problem was just how close it brought us to the apes. It kind of suggested that it wasn't that long ago that we were pretty ape-like. And a lot of people just couldn't buy this. And a number of major discoveries happened in Europe of Neanderthals. And the author that, or the principal investigator on that scene, does a lot of portrayals of these things and portrays them as extremely brutish, very ape-like, way more non-human than anybody had said so far. And that allows this guy, and actually the majority of scientists at the time, to say that there might be two lines. And you've got the Neanderthal Homo erectus line, and then somewhere way back in time, you're going to find the human line. But these two things have been separated for a very, very, very long time. And in fact, you'd actually expect, if you went back in time, for some kind of weird monkey to have got a big brain and then use that big brain to think its way into everything else, including bipedality and that kind of stuff. Actually, from our modern perspective, it sounds preposterous. But from the perspective of a group of people that were really not ready to digest this closest nature that we humans actually have, it was convenient. And it was so convenient, in fact, that the most famous fossil forgery of all time is perpetrated during this period, the Piltdown hoax, where somebody takes a human skull with an orangutan mandible and puts them together. Now, this is exactly what the majority within the community wanted. The majority of people were expecting that a human ancestor would combine the cranial, the brain size features of a modern human with a body that was more ape-like. And so the orangutan jaw and the modern human cranium fit right into what people were expecting. Now, this was primarily within the British establishment. The Piltdown was the only fossil known in Britain at that time, and they held on to it very dearly. In fact, it takes about 50 years for the lid to completely blow off this thing. Although discoveries that start to happen in Africa made people really wonder if this could possibly be the case, because none of them shared the same set of features. They didn't share this like big cranium and ape-like jaw. New species are named during this time, but of course, pretty much every new skull that's found is given a different species name, except the Neanderthals. And so we see sort of a proliferation of species names, and it happens at the same time that there are a lot of major discoveries made in the old world, and primarily in Indonesia, but also in Africa, as we'll see in a second. The stuff that is discovered in Africa is first sort of a Homo species, and then there is another group of fossils that are found in Indonesia at a place called Ngandong. These are Homo erectus crania, although they're just given a specific species name. It turns out that almost everything is given a species name at this time. Like I said, if you find something and it's different, it gets a species name. We're going to get a big self-correction on that thing, but we're now actually in another one of these times when we have lots of species names. And so uh, these were all sort of tried against the background of the fossils that were already had. Now, the first ape-man fossil, the Tong child, is found in the mid-1920s also, and this is actually ridiculed by the British establishment, who were so caught up in Piltdown and the implications of it that when a true missing link fossil that combined a small brain with human-like teeth, they wouldn't even look at it. Yeah, the Tong child's really an Australopithecus, and there's lots more fossils found from nearby that area that document this. But it's a classic example of how people's expectations shaped what they believed would come out of the ground, and everybody was wrong. We must learn that lesson in human origins. It's easy to make a hypothesis that is wrong. And here's a great example. This one is from Louis Leakey's big book from the 1920s and 30s. This is before he makes his big discoveries that we'll learn about next week. But at this juncture, Louis Leakey writes a book called Adam's Ancestors, and he maintains that deep division between the Neanderthals and their group and Homo sapiens. Now, when you read the paper for next week's writing assignment, this is actually part of it. This is actually something that comes to play, because one of the things that you'll see is that this line of thinking also leads to the perception of a very deep division between the different groups of people. And in the early 1900s, this was still a very, very popular notion that all the different groups of people had diverged a long time ago and had clearly been separated from each other. We know now that that's not the case at all, but look at this. I mean, you can see the way that he portrays these things when we zoom in on this part, and it shows you that they had an idea, or Louis Leakey and his contemporaries who were studying this notion of an earlier split between Neanderthals and humans were, well, they were kind of racist about things. That's the only way we would look at it now. They were racist. That was their interpretation, and part of their interpretation was based on their expectation. They expected wrong. It's a good example of how you have to keep an open mind when it comes to any kind of hypothesis of human origins. They're always loaded with stuff that is really, really meaningful in a socio-political way. And so you have to be careful, and we have to be very scientific. And that's one of the reasons why I'm such an ardent scientist, because when it comes to human origins, you just can't afford to be wrong. The consequences are too great, so we must be diligent about our science. Anyway, moving on. So while Louis Leakey is making crazy prognostications about the fossil record and everybody's caught up with all these brand new species, there's a revolution going on in biology. We already talked about it. The discovery of Drosophila and the discovery of all the subsequent discoveries that lead to a much better understanding of the way that populations work and speciations occur and all those things that happen from about 1905 to about 1925 and continue a little bit after that have a profound effect. They don't have an effect right away, but the things that are happening in evolutionary biology will have a big effect. And one of the biggest things that happens during the modern synthesis, which we talked about before, is a change in the way that people think about species. They stop thinking about species as these very simple allopatrically, like physically separated lineages. They go their separate ways as a result of that physical separation, and start thinking about them as complex populations and speciations that might occur either kind of on the fringe of an already existing species or even sometimes within an already existing species. These notions go a long way to transform the concept of species in biology, and it's this time that we see this real biological species concept of reproductive integrity starting to take a lead role. And actually, that species concept can be extrapolated over time. It's very difficult to recognize those boundaries in the fossil record, but we do expect that there will be these bounded forms, these populations, these groups that are bounded by reproduction that have some sort of trajectory into the past. And that's what we think of as an evolutionary species. It's a group of organisms that we expect to be reproducing, which we can't tell on the fossil record, that have similar evolutionary roles and tendencies. That is, they're doing the same thing and that they kind of look the same. This is a concept of a biological species, a reproductively defined species, pulled over a large amount of time, and that is an evolutionary species concept. And it's a really big difference from just this split and split and split. People think that when you look into the evolutionary past of any lineage, it's likely to be complicated, and there's likely to be changes in the evolutionary role over time. This notion that we could see species that way starts to become popular, and actually it's kind of going against what people are doing, naming all these new species in the early 20th century. One of the people most involved with this modern evolutionary synthesis was Ernst Mayr, and we see our first major revision of the human fossil record by a non-human paleontologist occur in 1950, when Ernst Mayr says, you guys with all your species and genus are just out of your minds. These are clearly things that could have reproduced with each other, and it's pretty likely that they did. And so to call something Homo soloensis and Anthropopithecus erectus and all these different genus and species names is probably not all that useful. And that notion stuck around for a while in the 1950s, and we'll see that it kind of dissipates a little bit. But also happening in the 50s is a discovery of radiometric dating. We've already talked about that one, and that's going to be really important. And so what I'd like you guys to do is pay special attention to the reading for next week's paper and do your best to think of that in the context of all the human origin stuff that we've been talking about today. And we'll continue this thread and continue talking about species and speciation on Tuesday. Now, you'll sort of see that we come full circle with this last slide showing Garnus discovering or developing the radiopotassium dating method in the 1950s. But as you'll see from doing your term paper reading over the weekend, they all go together, and this timing of the fossil record, applying time to it, is really fundamental when it comes to understanding not just human ancestor fossils, but actually human diversity. It comes at a time when people were seeing that the diversity in humans was most likely something that was related to them being separated for a long time. And we now know that a lot of that diversity on the outside is not really reflective of what has happened to us in terms of species. And it turns out that we are much more closely related to each other than anybody at that time knew. But this date that came out is going to help that, and I'll continue the thread explaining things in sort of a historical and biological context on Tuesday. With that, thanks very much for sitting through this long video and listening to it and being good students and everything like that.
shines. It shines, our soul, my friend. Then why can't we love each other? Said we should give together. Give me power to devour every hour. Give me power, oh Lord, to devour, oh yeah, every hour. I said, look what the Father has done. He hung our soul, my friend. Then why can't we love each other? Said we must give together. Give me power to devour every hour. Give me power, oh Lord, to devour, oh yeah, every hour. Give me power, oh Lord, to devour, oh yeah, every hour. I said I and I must live. We are waiting on our Father to come. Then man will know themselves. Give me power